Hey, Bio30s, and welcome to the start of the second half of the Reproduction and Prenatal Development Unit, Chapter 15 in the textbook, which is Human Development. The first half of the unit, we pretty much got to fertilization, where the two gametes met together to form the single-celled embryo, and now the second half of the unit is going to be what happens after that event occurs with the development of the embryo into the fetus and all the way to the birth of that organism. And of course, this unit, as we've been dealing with this entire unit, we're going to be focusing on human prenatal development, how it occurs in humans. Again, the chapter you're looking for, chapter 15, is going to be pages 506 to 543 in the textbook. And as always, you can use my website, boutasign.wixsite.com/mchs. Notes are there, assignments are there, videos are there. If you don't know this already by now, I don't know where you've been in any of my previous classes. All right. Let's get started. We're going to start a very simple little introduction today. We are going to recap fertilization, which I did touch on in the first half of the unit. So we're going to get to basically conception again, just to kind of refresh everyone's memories. And then starting in the next video, that's when we'll start kind of dealing with more of the actual development of what occurs after conception. All right, so first of all, we're going to be using a term a lot in this unit called gestation. Gestation is a term that is used to refer to the period of time that occurs between fertilization of a zygote and the birth of that organism, whether it's a human or an elephant or any organism that basically sexually reproduces, when it basically is going to, and again, we're dealing with mammals because they are gestating inside of a uterus, so placental mammals would be probably the largest group that we would associate with this term. In humans, gestational period is 39 weeks, which roughly works out to 9 months. That 39 weeks is divided into what are called 3 trimesters that are roughly 13 weeks each, or generally speaking, 3 months each. During this period, there is going to be 2 periods in the prenatal development, which would be the development of the zygote to the fully fledged organism that's born, in this case the human baby that is born at the end of the 9 months. That development, which we call prenatal development, is divided into 2 sections, which I will be getting to later in 2 separate videos. The first stage, which occurs from weeks 1 to 8, which would be during the first trimester exclusively, that's going to be the embryonic developmental period. That's where we actually refer to the human as an embryo between weeks 1 to 8. Weeks 9 to 39, then we're going to be going into the end of the first trimester and the second and the third trimesters. That is going to be called the fetal developmental stage, where we can actually refer to the developing human as a fetus. I will give you basically the details of both of those developmental periods in a later video coming up before the end of this unit. The next couple of slides, I'm going to skip over them, but if you want to read them, go ahead. A little takeoff of a Dr. Seuss kind of little storyline about birth. I usually read this out in class. I won't read this out to you now. You can definitely, if you're following along at home, give it a watch. It's kind of cute, but I'm not going to use this video time to read it out to you. We're not going to do story time today. What I would like to focus on in today's video, and again, it's a fairly short video today. I would like to kind of recap fertilization. We've discussed fertilization already, and we've dealt with the players already. I want to kind of recap what happened. So again, we've talked about our 2 gametes that have to fuse together. We've got our sperm and we've got our egg. The sperm obviously developed inside the male testis, inside the seminiferous tubules, developed into a spermatid. It then matured in the epididymis, continued to mature into the vas deferens, where it actually became fully motile with a full tail. Once it enters the ejaculatory duct, it's going to basically fuse with chemicals that are being released from the prostate, from the Cowper's gland, and from the seminal vesicles. That's going to basically produce our semen. During an ejaculation, that sperm would enter the female's reproductive system. It would start off in the vagina, head up through the cervix, through the uterus, and eventually, if lucky, gets all the way up into the oviduct. And in the oviduct, which would be the site of fertilization, it's going to run into an egg that has been ovulated by the female on day 14 of her menstrual cycle. And remember, there's only a 24 to 48 hour window for that egg. Once that egg is basically gone past a day and a half at most, basically not going to be viable. So there's a very short window where the sperm can actually, make contact with a viable egg. And interestingly enough, though, a sperm can last, once it gets to the oviduct, sperm can actually last in the oviduct for up to 3 days. So they can hang around if they're there maybe a little bit before ovulation occurs. They can be sort of lying in wait for that egg to be released from the oviduct. And remember, the egg is not technically an ovum. The term ovum is used quite often to describe a female, a scientific term to describe the female egg. It is not released as an ovum. When it's released from the ovary during ovulation, that egg is referred to as a secondary oocyte. It will not become an ovum and eventually a zygote until it actually becomes penetrated, or actually gets entered by a sperm, or a sperm enters it, I should say. So let me kind of explain how that happens. So we talked about the anatomy of the sperm, and we discussed how the sperm had on its head, it's got this little cap known as an acrosome, if you might remember from my discussion on the male reproductive system. That acrosome contains a bunch of enzymes, and these enzymes are designed to actually penetrate the outer casing of the egg. Now remember, the egg is extremely large in comparison to the sperm. I've mentioned this before, but if a sperm was the size of a human, the egg would be about the size of a 30-story skyscraper. So a very large cell. So there might be multiple sperm, dozens of sperm, if not hundreds of sperm that have maybe survived, that are now maybe trying to access that egg. Only one of them is going to be allowed in. The sperm, though, is going to release its digestive enzymes. It's going to crack open its acrosome in the head of the sperm, and that's going to allow that sperm to begin to start to kind of tackle the outer layers of the secondary oocyte. The secondary oocyte has 2 layers outside the cell membrane that you should be familiar with. The outermost layer is called a corona radia, no relation to coronavirus, but just the corona radia. And then the middle layer is called the zona pellucida. I like to call it, Lollapalooza. I always think of Lollapalooza when I think of this. So zona pellucida. It's a great musical festival somewhere in the oviduct of a female. I don't know. But these are the 2 layers that the sperm actually has to digest through using the digestive enzymes of the acrosome. And then once the sperm enters it basically fuses with receptors on the cell membrane. And then when that happens, basically the outer layer of the egg locks off. No other sperm that are basically there are going to be able to access the egg at that point. It basically locks itself off to the one sperm. That sperm then will basically infuse its genetic material into the secondary oocyte. And that's going to set off a chain reaction of events. That genetic material is going to have to sort of drift its way in toward the center of the egg. It's going to get carried toward the nucleus. There is some time while this is occurring, and a few things are going to happen while that's happening. So the secondary oocyte, once the sperm enters, or the genetic material enters the egg from the sperm, it's going to have to finish meiosis too, because actually the secondary oocyte has only undergone its form due to meiosis 1. We haven't undergone the second round of meiotic division yet. So the second round of meiotic division will occur when this happens. This is going to cause the secondary oocyte to become an ootid. The ootid will then very rapidly mature into the ovum. Now remember, when a sperm was a spermatid and became a spermatozoa, there's a long period as it going through the epididymis and through the vas deferens for this to occur. In an egg, that maturation period occurs in a very, very short amount of time. Again, it's got to happen before the sperm's genetic material basically comes in contact with the nucleus of the egg. At that point, when the 2 genetic materials, the 2 haploid pieces of genetic material come together from the male and the female, that's when you actually get fertilization occurring. You get the fusing of the genetic material to a diploid genetic makeup. And then at that point, the single cell at that point is now going to be referred to as a zygote, because it is now a diploid cell rather than the original haploid configuration that both of these cells were before fertilization. It's also at this point that chromosomal sex is determined. Obviously, the sperm is going to determine whether it's a male or a female. If the male's sperm has a Y chromosome in it, it's going to be, obviously, the zygote will become chromosomally a male. And then if the male's sperm has a X chromosome that is providing, then obviously the zygote would be chromosomally a female because it would be an XX configuration. Now we are going to discuss some chromosomal variations of that due to non-disjunction. Later in the unit, there are some disorders out there known, like a Turner syndrome would be an example of one, where you're getting either trisomies or monomacies that are occurring when you get the fusing of the egg and the sperm. And we'll discuss that a little bit later in the unit. But for now, just be aware that chromosomal sex, chromosomally speaking, the sex of the embryo, or the zygote at this point, is determined at the moment of conception due to the configuration of the chromosomal, the sex chromosomes, and how they actually arrange at that point. Diagram here is sort of what I've drawn on the board, a little bit more detailed than what I've drawn, but this is a great little diagram that shows you the chain of events that occur as the sperm comes in contact with the outside of the secondary oocyte. At this point, you can have, sometimes you can have scenarios that occur that allow for the birth of more than one organism or more than one human at a time. You can get twins. I mean, there are people out there that are twins, and we have twins here at MCHS, and there are two types of twins that can form during fertilization. There are what are called dizygotic twins and monozygotic twins. Now, the name di and mono would imply that we've got two zygotes here. We've got one zygote here, and that's exactly what the name implies. A dizygotic twin basically refers to when a female has a multiple ovulational event. She doesn't release one secondary oocyte. She releases two secondary oocytes at the same time. They could be coming out of presumably two different oviducts. The sperm that have entered into the female's reproductive system obviously have a choice. When they hit the uterus, they can either go to the oviduct on the right or the oviduct on the left. If they're going to the wrong oviduct, it isn't ovulating, then there's going to be no egg for them. But if a female has a multiple ovulation, there could be secondary oocytes coming down both the oviducts. Is you get two separate fertilization events to two separate eggs. So you get two distinctive zygotes that are basically now going to enter the uterus and that are going to implant in the uterus roughly on day 21 of the female's menstrual cycle, just when the endometrium is at its thickest. And then basically you're going to have two separate zygotes, completely different from one another, but we would refer to them as non-identical twins or as fraternal twins, as you may have heard. If you have a single zygote that gets that gets fertilized normally, but if the first division off that zygote divides into essentially two zygotes rather than a zygote with two cells, then what you get is you get a monozygotic twin. You get twins. You get identical twins, and this would be the rarer of the two events because there has to be sort of a divisional error on the first division. When the zygote becomes a two-celled embryo, instead of becoming a two-celled embryo, it becomes essentially two zygotes that are essentially mitotic twins of each other because it's a mitotic division. So it'd be like almost like a bacteria cell divided into two identical copies of itself. Now that doesn't happen very often, otherwise there'd be lots of identical twins out there, right? So this does occur occasionally. There's sort of an error that occurs, but it does lead to identical offspring, genetically identical offspring. Now, sometimes that division can also have an error in it. Not that, and I said it was an error, but I mean it's something it's an abnormality, I guess, or an anomaly that occurs with the division of that one zygote into two zygotes. Well, if there's an error in that anomaly, whereas let's say the two zygotes begin to split from one another, but they don't split completely. Cytokinesis, for some reason, doesn't completely separate the two cells. There's a joining of those two zygotes where there's a little section of the cell membrane that stays attached for some reason. You can wind up with what are called conjoined twins. Not appropriately named Siamese twins. You may have heard the term Siamese twins. That's not politically correct. The reason why they're called Siamese twins is the first very famous conjoined twins that kind of got really well known around the world were a couple of twins, Chan and Eng Bunker, who were brothers from Siam, which is now Thailand. So the name Siamese twins sort of stuck. They actually became a very famous circus act around the world. They traveled with the famous circus gentleman by the name of P.T. Barnum. And as you can see in the diagrams, they were actually only connected by a very thin piece of skin between their abdomen. And they could have actually been separated. Even in the 1800s, the process probably wouldn't have been that difficult to separate them. But their livelihood was to be conjoined twins. So they basically decided not to get separated and basically made a career or a living out of becoming this kind of circus act that basically traveled around the world, and they became very famous. There's been lots of other conjoined twins over the years. You may have heard of Abigail and Brittany Hansel, that are the famous conjoined twins from the United States. They were born in 1990. They have a single lower body with two upper bodies. They have two heads on the top. And if you Google them, they had their own, even their own television show for a while. They were on a reality television show. But they basically have they have two hearts, two esophagi, four lungs. They have only one rib cage. They have their own central nervous systems, their own brains, two gallbladders, two stomachs, three kidneys, and then as you go further down, they have one liver. They've got one large intestine, one small intestine, one pelvis, and one set of reproductive organs. So they have only one set of gonads for both the twins. So a very interesting split there, and they're both, they're both. There's no developmental issues as far as cognitive issues or anything like that. They're right now they're in their 30s at this point. They've been living their lives out, but again they are now literally joined at the hip, as it were. There's other examples, like Lakshima Tatma was born in 2005. She basically had at the bottom, her and her twin were fused at the bottom on the ends of their spinal cords, on their coccyx, on their tailbones, and the one twin at the bottom did not fully develop, as you can see in that diagram there. And she was actually named after a Hindu goddess, and there was actually people that came to her village believing that she was a reincarnation of a multi-limbed Hindu goddess. She did actually get separated from her parasitic twin because her parasitic twin didn't have a head, so she just had to be separated from her. The operation lasted about 27 hours. They had to remove the parasite's abdominal organs. They had to remove there was a kidney that wasn't working that they had to remove. They had to move the reproductive system and the urinary bladder of the one twin that would survive the procedure. And they had to amputate the parasite's legs, hip joint, cutting the joint backbones. They had to separate the two backbones. Had to be very careful that they didn't paralyze the actual, Lakshima. That they didn't paralyze her, and so there was a lot of work involved there. So they were separated actually at the tailbone. And then there's another example here, and this is actually not fairly common, but it's one of the more well-known types of conjoined twins when they're joined at the skull. There's a couple of brothers that were born 2001 in Egypt. They had basically a 1 in 2 million chance of basically being conjoined at the head. And they had to go to Dallas. Dallas has become very well renowned. They have a great surgical center in Dallas that specializes in the separation of conjoined twins. The parents were given the odds that both their twins survived was only about 10%, but they felt that them being joined at the head like that, essentially they had two brains, but they had to figure out how to separate the heads respectively to kind of see if they could separate both the twins. They did proceed with they went with the procedure, even though there was only a 10% chance that both the twins would survive. The one twin, I think, was given a better chance than the other of survival. 34 hours to separate them, and actually both the boys managed to survive the procedure and are now and are still alive. They've been now they would be in their 20s by now. They had some cognitive delays and issues because of the nature of having them to separate their heads. There were a few cognitive issues that occurred, but overall their health is actually quite good, and they've been able to now lead fairly normal lives, as opposed to, imagine if you were stuck two heads together, it would not be a normal life by any means. That's our conversation for today. Just an introduction into prenatal development and fertilization, and some of the terms about embryonic and fetal development. No homework for today, just hopefully you took a couple of notes on what happened today, knowing the difference between dizygotic and monozygotic twins as well. Not a bad thing, conjoined twins, that kind of stuff. Next day, we are going to discuss ARTs, which are assistive reproductive technologies. How do people use technology to help get themselves pregnant if they're having issues with infertility? And also I'm going to be going over methods of contraception, which ones are the most effective, which ones are the best at preventing, let's say, things like STIs, I'll be discussing that in my next video. Until then, thanks for tuning in, and we will see you next time.